Hello and welcome to the pharmaphorum podcast. I'm your host, Jonah Comstock. My guest today is Vicki Richon, CEO of Entact Bio. Entact Bio is doing something different in small molecule space. Whereas most drugs focus on inhibiting harmful proteins, Entact focuses on enhancing helpful proteins. I thought this was a pretty interesting novel idea that I hadn't heard before, so I'm eager to learn more about it along with you. Welcome to the show, Vicki.
Thank you so much. I'm really happy to be talking to you today.
So before we jump into Entact, I know you've got quite an interesting career in pharma. You've really done a lot, and you've been involved in big pharma companies. So tell me a little bit about how you got here and what's your story.
Sure, happy to. So I'm a scientist by training, and I've spent really half of my career in academia and the other half of my career now in more industry and drug development and industry. So a scientist, as I said, by training. I did my early career in chemistry and biochemistry, but then decided I really wanted to do something very translational early in my career that was going to have impact on patients, which now seems like very ordinary, but many years ago that wasn't something that was really thought of very much. And so I decided to go to Sloan Kettering Cancer Center in New York, and that's where I spent most of my time in academia. And while I was there, though, I was able to work on a project that we actually took to patients in the clinic, and we developed a drug that was targeting epigenetics. And the idea is that we would be able to induce differentiation of cancer cells rather than killing cancer cells. So a theme throughout my career is that I like to work in new areas and bring new types of drugs to patients. I had the unique opportunity there to write all the regulatory documentation and take that drug to patients while I was doing research at Sloan Kettering. That really transformed me to want to take really great science and apply it to drug discovery. We started treating patients, and we saw patients responding to the drug. So we decided to actually start a company. And so I co-founded a biotech company with others and left Sloan to actually continue development there. The drug was looking very successful. The name of the drug at the time was the generic name Verinostat, which after it got approved became Xalinza. But I'm getting ahead of myself here. So we started the company, and things were going really well. And so we got acquired by a pharmaceutical company, Merck. And so I then had the great fortune to go to Merck and continue the development of the drug, and have another part of my career where I really was embedded in drug development with a pharmaceutical company, which is really great training for people. Then after several years of being at Merck, and as I said, the drug was approved as Xalinza, I wanted to go back and apply all my learnings to biotech again. And so now I've done that. I went to another company called Epizyme and was there at the very beginning and helped build the company, but then wanted to go back again. I had an opportunity to go back to another pharmaceutical company, Sanofi. And so I went there for several years, heading up the oncology, drug discovery, and translational medicine, and then flip-flopped back to leaving there after several years and going to build a company. And now I think I'm in my path and staying in small biotech. So I built and was CEO of another company called Ribon Therapeutics. And after Ribon, we moved several compounds into the clinic. I decided I wanted to go back to the beginning and build again. And that's when I left and really found that I was really intrigued with this idea, as you so nicely introduced, that I had spent the whole previous part of my career really inhibiting proteins in cells that were overactive in disease. Whereas we know now that we've been able to study really and understand disease, that there are many, many diseases that are actually caused by too little of a protein being present. And yeah, and so but we don't have many ways of fixing that. Like there's been genetic methods such as gene therapy that we've heard a lot about, where if the protein is missing or the gene is defective and missing, can you put a new gene in? But there aren't that many small molecule approaches to being able to basically enhance a protein that's just there in too little of an amount. So that's what we're doing at Entact.
How does that work? Because when you're inhibiting a protein, a lot of times what you're doing is you're sort of blocking the antigen. So there's a connection that's supposed to fit together, and you keep it from fitting together. But what does it mean to enhance a protein?
Yeah, no, that's a really, really great question. So, what we mean by enhancing a protein is that you can increase the amount of that protein or change the activity that's gone wrong in disease. And so, what we're combining is this knowledge that in disease there's too little of a particular protein with another new technology that actually has been developed to, again, inhibit proteins. So let me make sure that this is understood. And so one really new wave of small molecule drug discovery is to have a small molecule that can actually bind to your protein of interest, but also then bind to another protein that's going to modify your protein of interest. And so there's a whole field called induced proximity. So you have sort of a small molecule that has two ends has two different functional ends combined with what's called a linker. And so what's been done is in this field that's become pretty acceptable now and highly used is called PROTACs. So PROTACs actually take your protein of interest and bring in proximity another protein that adds a small protein tag to your protein, then that causes that protein to be degraded and gone. So it's a very interesting technology that's called the degrader technology. So what we did was we took that technology, and what we now do is have we have our target protein, and we bring in proximity another protein that removes that tag for destruction. So now you have less destruction of your protein and you have more protein.
Yeah, that makes sense to me. That's really interesting. So can you let's try and make it a little more concrete. Are there any examples you can give of like a condition where this is really promising, and what makes it promising there, a therapeutic area or a use case?
Yeah, so I can give you one case and a couple different examples, but the first one I'll start with is a devastating monogenic disease called cystic fibrosis. So in cystic fibrosis, there's too little of a protein called CFTR. And see, that's the only defect in patients that have cystic fibrosis. And because there's a mutation in that protein, it doesn't fold correctly and it gets targeted for destruction because our bodies say, hey, there's something wrong with this protein. I'm going to destroy it because I don't want it around. And so it tags it with this ubiquitin or the small molecule tag called ubiquitin, and that sends it for destruction. That disease now has been pretty much cured because we have small molecules that can actually fix that and and what's called potentiate it and increase the amount of that protein. We've been able to show with our technology that if we have that small molecule bind to it, but now add this other end to the molecule that brings in the enzyme that removes the ubiquitin or tag, that protein now is increased in abundance. But what's really cool is that this is a proof of concept for us because this disease has been shown that if you can just increase the amount of this protein, you now have changed the lifespan over the last 20 years or so from being a 20-year-old to now a 65-year-old.
Wow.
So just increasing the amount of that protein can cause this change in longevity in people.
One of the things I always ask when someone has an approach that's so kind of new or paradigm changing is why is it that it's taken this long for people to realize this? Why has there been so much focus on inhibiting and not as much on enhancing?
Yeah, it's not for want of trying. It's usually what's unusual about the cystic fibrosis example, is it's very hard to find small molecules that can bind and not only sort of potentiate the protein, but kind of fix the protein. So what's new is that we just need something to bind, but it doesn't need to do anything because the second protein is going to come and be able to actually remove that ubiquitin tag and cause the increase in the protein. So what's also new is this whole field of induced proximity and PROTACs. So PROTACs have been around now for about a decade or so, and they, maybe a little longer. But the first PROTACs that cause induced degradation of proteins are now going through in late stage clinical trials, and we're anticipating that within the next year, we're going to have the first drug approved that's using the opposite end in the destroying proteins, but the same technology.
And that technology is foundational to be able to do what you do.
Exactly, exactly.
So, what do you think the impact of this technology in the long term might be? Like how widespread are the possibilities, and how do you see it kind of panning out in the future?
Yeah, I think whenever you add a new type of modality, it can have really amazing impact. So I look at it maybe as the early days of having antibodies as therapies. We didn't have antibodies as therapies, before the last basically 20, 30 years. And so now that we have antibodies available, that's just given the doctors more medicines to be able to treat people with. So I think that this will really open up a whole new avenue for doctors to use and to treat patients. So just let me give you an example. One of the things that really intrigued me before I came was thinking about how many different types of targets. I gave you one example of a protein that's decreased in disease that if we amplify it, we know, increase it, we know there's been a therapeutic effect. How many more of them are there? So we found about over 2,000 different proteins that are disease related, that are decreased in disease, that could have relevance with our technology. And so that's a lot of different diseases that could be impacted and patients that could be impacted. And I think that's what's really exciting to me about this opportunity. Another example of a specific example of where this technology could be used is in cancer, because cancer we know a lot about, and where we have really focused our therapies is in several different areas, but one that's quite common within the cancer cell itself, that there's what's called oncogenes, which are cause cancer. And but then there's the flip side of tumor suppressors, which are lost in cancer or decreased in cancer, that are also basically happen in every single cancer. And so we've been had a lot of success in drug development again in inhibiting oncogenes. And that's where a lot of these new therapies have come from recently in terms of being able to have real impact in patients. But we haven't made very much progress at all in the other fundamental defect in cancer, and that's tumor suppressors that are lost. So if you can increase the amount of this protein, you can suppress the formation of cancer. And so this technology, we believe, will have, fundamental importance in being able to increase those tumor suppressors as well.
So, I suspect that it's not a one or the other kind of situation, right? I mean, can you get potentially even better results by both suppressing and enhancing?
That's right. Like suppressing, inhibiting what's bad and enhancing what's good within the cancer. So we think that that's absolutely the case. And certainly in cancer, combination therapies is what is really guides most therapeutic approaches. So we think this could add, though, a really nice element that hasn't been able to be done before. Whereas for most of my career, we've been trying different approaches to be able to do this. So I think this new modality has a true potential to be able to increase those tumor suppressors.
That's all really interesting. I'm going to jump back, and I hope I'll be able to wind this back around again. But when you were talking about your bio, your background, I just thought it was so interesting that you've been in, clinical academic research and then kind of ping pong back and forth between big pharma and small biotechs. And I'm curious, like, what about those experiences made you decide that kind of small biotech is where you want to land? Are there things about that environment that make it easier to do this kind of innovative work, or what are the pros and cons? I'm just curious, sort of how.
Yeah, I ping pong back and forth because I think both have huge opportunities and value. And I think they've really taken me to where I am today. So, in large pharma, what you get to see is many different projects and work with large teams of people. So, you can see many, many different things at the same time. So, you learn a lot about different projects, what goes well, what goes wrong, how to work as a team. Those are some of the things that I really learned at Merck that really equipped me to be able to go into the small company where you're focused on fewer things, but usually have a broader view of them. Certainly the same thing happened when I went back to large pharma to go to Sanofi. I felt like I really learned there. I had a group of when I started there, of about 350 people. So I really learned about leadership. I learned about also being a leader within the company, um, really how to sell ideas. It might be surprising to people, but even in large pharma, you have to defend what your group is doing and convince the rest of the organization that what you're doing should be prioritized and done. And so that's all really important. So I felt like I had a lot of skills to be able to go to small companies. And build the small company and be able to be a good leader and to understand what is important and how to really lead. So I think that both are really great. They're different, but they're both really important for doing drug development. And for me, they were great opportunities to really build my skill level to be able to really be a good leader, try to be the best leader I can in biotech.
I feel like we've entered this era where the small biotechs are such an important part of the innovation ecosystem, and big pharma are really relying on them to do the innovation that's a little more out there, a little bit riskier. But at the same time, being a small biotech, even when it's hard, even when your idea is really good and your science is really good, you've got to make sure you can manage your capital, you can sell that story, and that ultimately you can find some way to either license or be acquired or produce yourself and get that drug into patients' hands. So it's it's an interesting kind of like number of things to balance on your plate. And I'm curious, like, how you think about when you're thinking about the kind of the long term and the future for this company, that running that marathon, not a sprint, and making sure that you really you do the science right, you get the approvals, and you keep your runway. Yeah, talk to me a little bit about that experience.
Yeah, I think, one of the things that you really learn, that I learned by being now a second time CEO and being on from the research side, is how important it is to make sure that you have alignment with regarding the strategy, just as you said, for the company, and that you align with your investors and your board to make sure that you all have the same vision for the company and put in that right financial plan that matches your research plan. So I find that I really think about it in a way of sort of what do you need to accomplish in the first year, in the third year, and the fifth year. What does that plan look like? And I think the first year, usually, when for a company like ours, where we really have an idea and we need to be able to show that that idea works. And so in the beginning, it's all about assembling a great team and driving to having that early proof of concept for your technology. And then by the third year, you really need to not only platform focused, as we're a platform company, but also asset focused, that you're bringing assets that are driving value forward. And then by year 5, my goal is to provide as much optionality for the company as possible so that we might make the right financial decisions as we go forward. But I've created optionality, for the company in terms of having pharma partnerships or being able to go public or, having an M&A opportunity, be acquired type of opportunity, so that I've created as much optionality for the company to be successful as possible. So that's how I think about the early plan for a company.
And where are you in the journey right now with Entact? I mean, I know you've got some things in the pipeline. Where are you in terms of like phases and what's kind of the roadmap?
Yeah, so we're still a little on the early side, so we're still very stealth. But we've been starting to build our pipeline right now. We haven't discussed yet what the projects are in a non-confidentially, but I'm really excited about where we are in terms of building the pipeline and starting to really show proof of concept for this approach with our molecules. And so really excited about where we are and the potential for this and to continue to build the company.
Yeah, I mean, you said that the foundational technology that this is built on, those protacts, are fairly new, right? And they've only sort of recently. So you're kind of right on the front edge of this. You can't really run any faster than that, right?
So we often describe it as, building the plane as you're flying it. And so that's definitely what's exciting about where we are, is that we're really on the forefront. So PROTACs are the technology to inhibit proteins, and our Intacts are the technology to enhance proteins. And so we're taking the learnings from the PROTACs, but we're also learning where they actually fall, since it is a different modality than the PROTACs, as to what's going to be the same and what's going to be different. So we're creating the roadmap for what we're doing as we're doing it. And I think that's what's really exciting, is to every day be getting new data and driving innovation.
Is it hard to be in small molecule right now? You hear sometimes that more of the money, more of the attention, more of the excitement is around biologics, is around cell and gene. There was, the kind of, you might call it a snub of small molecules with the IRA. Like, do you feel like it's being overlooked right now, or?
I think that if you actually look at the number of deals that are still being done and the value in drugs that are being developed, that while there is, it's great that there's a diversity again, because you need to attack each problem in a different way. The mainstay, I think, are still small molecules. And so but now this new type of small molecule, right, that induces proximity. And so that's another type of small molecule. That's a next generation small molecule. So I think that, but certainly, like I said, I think it still will become a mainstay of what we're doing and will be there. But I think it's great that there are other technologies that can answer other problems. Certainly, antibodies have made a huge difference. And in oncology now, antibody drug conjugates have become very popular, but I think we still have a lot to do and a lot of work to do to actually have cures.
Talk to me a little bit about what motivates you from, thinking about the patient and the end user for, some of these technologies. You talked a little bit about cerebral palsy, and I know that's a really challenging condition. How do you kind of keep that in mind in your work at your company? And yeah, talk to me about that piece.
Yeah, so patients are in our mission statement, and helping patients and providing new therapies are from our mission statement to how we think about developing programs. We really think first about the patient and the disease, and then say, what does our technology apply? Where can we solve problems in disease where there's too little protein? And so the cool part about that is that we start our program with a disease and patient in mind, and patient population in mind. And then we understand whether our technology can fix that problem for that patient, and then develop the drug as we go forward in the next few years. But then we have, at the end of that development of the drug, before we go into patients, we know what patients to go into because we've started with the patient. So a lot of people say bench to bedside. I like saying bedside to bench, and then back to bedside again, and back to the patient. And so I think that's really how we think about drug development at Intact.
That's wonderful. You mentioned you're a platform company, and that makes sense to me, knowing what I know about kind of where the innovation is. But what does that really mean? Like, how much work will the platform do, and how much work will you have to do in each iteration? If that makes sense.
Right. So the idea is really that the platform will support the programs and the drug development for the programs. And so once we establish the platform, which is what we did in the first few years of the company, is that we now have developed the technology and the assays and everything you need to do drug discovery. So our first program that we're applying this to is sort of the test case, if you will. It's not really the test case because it's a real program, but it's the first one that we're running through. And the second program will be easier, and we'll be able to apply the technology that we developed for the first program and within our platform to the next program, to the next program, to the next program. And it's like learning all for the RNA companies, for example, learning all how to make an RNA, how to make it less immunogenic. So that's what we're learning, how to make our technology work. What type of linkers do we need? How do we build the molecule? What assays do we need? And so we've built that technology, and our what we call our platform is Encompass. So we have Encompass 1.0 that we're now employing in our programs. And as we continue to learn, we'll continue to improve the platform, but we'll be able to use all of the assays and developments we have within the platform to apply to programs. So we have three real components to our platform. And the first one is really understanding target selection and how we select targets that are going to be amenable to our approach. The second component of the platform is really what protein, as I called before, we call the enzyme that we're recruiting to our target protein a D-ubiquitinase or DUB. And so we have understood the DUBs and ubiquitin. And so we've been able to select the DUBs and which DUB for the protein of interest. So we have that understanding now within our platform, and we know how to recruit a DUB to a specific protein. And so that's the second component of our platform. I will also say that one of the other things I'm really proud about at Entact is that we have a scientific advisory board that are experts in ubiquitin and DUBs. And so they have over 100 years of experience in DUBs, and from biology to biochemistry to signaling with these enzymes, chemistry. And so we've taken their knowledge and worked very closely with them to apply it and combine with our drug discovery knowledge. And that's really powerful approach. And then the third component of our platform is putting the whole molecule together and having the structural insights to be able to say, I know where to target on this protein versus where to target on our target protein versus the DUB, and how to bring the two together. So it's like which target, which DUB, and then the molecule itself to bring them together. So those are the three components of our platform that we've developed that, as we said, really make each program now go much faster and quicker.
And each time you complete a program, you'll learn even more about refining the platform. Yeah.
Exactly.
We're coming near the end of our time. Is there anything I haven't asked you about that you really want to talk about, or you think is important about the work you're doing?
I just want to perhaps, I think we covered a lot and a lot of really great things. I probably just want to emphasize a couple of things, in terms of the real potential and vast opportunity for our technology. And that's what's really exciting to me, is that we know from having all of our sequencing data done and everything that everyone has learned about disease in the last decade or so, where we know that there are just so many diseases that are still untreated. And so many of those diseases are caused by a decrease in a protein. And this technology really has the potential to be able to be a new approach for those patients that have those diseases. So I'm really excited about that opportunity, and the team that we've developed have here at Entact to be able to approach that in a very systematic way. Because what we're doing that I think is very exciting is that we're combining really being able to select that specific protein and understand now in disease that protein that's changed and alter that protein and enhance its activity to treat that patient.
Well, thank you so much for chatting with me. I think it's very, very interesting what you're doing, and I'm looking forward to hearing more as you have milestones to report.
Great. Thank you. It was a real pleasure to talk to you today, too. Thanks, Jonah.
That concludes this episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about other installments in the series at pharmaphorum.com/podcast. The pharmaphorum podcast is also available on iTunes, Spotify, Acast, Stitcher, and Podme, where you can find and subscribe by searching for pharmaphorum. And don't forget to visit our website, where you can sign up for daily news and analysis bulletins, and to follow us on Twitter at pharmaphorum. Thanks for listening.